with Francis Crick, François Jacob and Jacques Monod discovered messenger RNA. In 1961, Marshall Nirenberg decoded the genetic code. Actually, in 1961, he decoded the first triplet, and over the next 5 years would decode more than 60 codons. So what they used was a cell-free system, to which they added, they incubated an RNA sequence that was so they tried a lot of stuff, but at one point they tried a sequence made only of uracil, which ended up generating a very, very long chain of phenylalanine, for which the code is UUU. So they had identified the first codon, and he would use some similar method to identify the rest. In the 70s, Hamilton and Smith would discover restriction enzymes, which turned out to be extremely important for cloning. He also won a Nobel Prize. Paul Berg also won a Nobel Prize for creating the first recombinant DNA molecule, but he did not try to insert it in cells. This was done the next year by Herbert Boyer and Stanley Cohen, who showed that DNA could be cloned in cells. On this page, they're the only ones who did not win a Nobel Prize. I can't speak for Stanley Cohen, but don't feel too bad for Herbert Boyer, who ended up founding Genentech and made several tens of millions of dollars with that company, who actually was the very first company to use genetics commercially. If we step a little bit forward with DNA sequencing, that was another seminal moment, which is still extremely important today. So the same year, two different techniques were developed. One was the chemical sequencing by Gilbert and Maxim, for which Gilbert won the Nobel Prize. And the Sanger method, or chain termination method, is still in use today. Whereas the Gilbert method was a little bit more complex technically and used toxic reagents. So the Sanger method took much of the market. So if we skip the chemical sequencing, the Sanger method, most of you are probably familiar with that. So they would synthesize DNA from single-stranded DNA. And then they would use 4 different mixes, which is something that we don't do today because we have alternative technology. But they would use 4 separate mixes with deoxy. In each and every mix, there would be a D-deoxybase that would interrupt amplification. And then you would do the electrophoresis on a gel, and where it would give rise to different fragments of different sizes. With the size where the stop codon is would be in the lane of a given base. Eventually, this was adapted to fluorescent bases by Applied, and you still see these reads if you do a lot of cloning. You get these reads regularly, and was applied to automated sequencing in 1986. And then we entered the era of linkage analysis. So in 1978, the first polymorphic markers in the genome were identified, which were the RFLP restriction fragment length polymorphisms, which are actually sites of cleavage by several restriction enzymes. Using these, James Casella was able to link Huntington's disease, which is a very rare but fairly frequent autosomal dominant neurological disease, to a polymorphic marker on chromosome 4. But they were not able to identify the gene associated with that marker until 10 years later, in 1993, I think. Obviously, 1983 was also the year that PCR was invented by Kary Mullis, which was also a revolution in molecular biology, facilitating you did not need to amplify your DNA in VAX anymore. And 6 years later, in 1989, The first gene, the first disease gene was identified, the gene for cystic fibrosis. So we're now 26 years later. And the very same year, a second kind of polymorphic marker, I don't know if this is the second one, but a significant kind of polymorphic markers was identified as microsatellite markers. And I just want to spend a few minutes discussing that because it has been significant in the way we can do linkage over the past, at least during the 90s and early 2000s. So genetic polymorphic markers are DNA sequences that have known now. Well, at first they had to map them, but now we know that where they are found in the genome. And there are several types, probably close to 10 types. But the first one was the RFLP, which was the first one also used for linkage. But upon the event of microsatellites, they took over for RFLP because they had several advantages. You could find, in the end, I think they identified about 10,000 of them in the genome. They have very high heterozygosity, which means that whereas the RFLP don't tend to vary a lot over time, microsatellites have varied a lot. So they don't vary much from one generation to another, but it gives you a very high level of heterozygosity, partly because they are multi-allelic. So you can find, so they're very variable, and you can find several alleles for them. Technically, it's extremely simple, especially in the era of PCR, where you just have to amplify and look at the size. And they were able to allow genome scans at a resolution of about 10 centimorgans. And more recently, with the sequence of the human genome, another type of polymorphic markers, which are SNPs, and many of you are probably familiar with that, appeared. One of their advantages is that they are extremely frequent, about 1 every 1,000 bases, which allows to do a very high resolution genome scan fairly easily. Unfortunately, they are diallelic, so they don't have as many alleles as the microsatellite markers. And this is just one example of what it would look like to do linkage using the microsatellite markers. So you know where the marker sits on the chromosome, and there are variables. So in this case, it's a CA sequence, which can have, I think it's 14, 15, 16, 17, or 18 repeats. Then you would use the same you would use primers to amplify that. It would give you fragments of different sizes depending on the number of repeats. So in this case here, you have the mother who carries on one allele with 18 repeats and one with 14. You have the father who has the disease, who has one of 15 and one of 16. And you have the son who also has the disease, who has one of 15 and one of 14. Obviously, this one is from his mother, and this one is from the father. Obviously, one would have, before we were, because you would look at any region of the genome, any chromosome, you would find something that you would find the same chromosome in the parent, in the son and in the father. So before we associate this marker with the disease, one would have to build a haplotype. So look at several markers and try and see if the whole region is shared. And that it would be extremely hard to prove that, except for a very, very tiny region, that this is associated with the disease. One would be helped if you had several other families where you would find the same haplotype. Still in the era of molecular genetics, we now get to 1989 when Eric Lander and Botstein would describe how you could build maps for quantitative loci. So the interval maps for quantitative loci. And the quantitative loci are traits that are not are on a bell curve. So these are complex traits, like size, the color of the eyes, the color of the skin. They're often associated with several different genes. So this is the main method that was used later to study those traits. 1990, the Human Genome Project was launched. People were expecting it to last for 15 years at a cost of $3 billion. And we'll get to that in a second. So in 1990, it was the first try and the first successful attempt at gene therapy for an immune deficiency. Unfortunately, in the 25 years since, successes in gene therapy are very few and far between, which, especially in neurological disorders, probably has to do with delivery. In 1991, Craig Venter, this man here, described a way to detect genes using express sequence tags. So the ESTs, not used much today because we have the whole sequence, but people had no idea what, and it was very hard to figure out what the sequence, what the genomic sequence was, and what was significant, which were genes, and what was introns, and what was at the time junk DNA. So express sequence tags were basically mRNA reverse transcribed in cDNA, and one would expect that if it's expressed, it is probably significant. So it allowed for the identification of several genes much more quickly. 1992 was a description of comparative genomic hybridization, and I'll show you an example of that in a few minutes. And in 1995, so the first complete genome of an organism was the first complete genome was sequenced was for the bacteria Haemophilus influenzae, using the shotgun approach, still by Craig Venter. And we have to get into that for a second because at the time of the Human Genome Project, these strategies were in opposition. So there was the Human Genome Project who wanted to go very slowly using the golden tiling path, where they would cleave the DNA in 150 megabases, really position that, know where these 150 megabases are before shearing the DNA, and then sequencing, doing that in a very orderly fashion. Venter said, well, let's get rid of the back, let's just shear the DNA and sequence directly, which is the shotgun approach. So before everything, he had to prove that this technique worked in simple organisms, such as the H. influenzae, in which they were able to sequence. And it was also important because the Human Genome Project was public, whereas Venter's project was private, and they wanted to patent a lot of the genes. So it really was a very big incentive on the public project to be more efficient and faster. So in 1998, the first multicellular organism was completed. That was not done by Venter's group, but by a consortium using the golden tiling path. And they were able to announce that the sequencing was completed in 2000. That they were done in 2000, but really formally completed in 2003. So, as I said previously, they had predicted that it would be done by 2005, so it was 2 years early, and they thought it would be $3 billion, $2.7 billion, 300 million is not an insignificant amount of money. In 2005 was the first genome-wide association study. I won't speak much about that. These are the kinds of studies that target complex diseases. Complex diseases. So I'll just give you an idea that depending on the allele frequency for Mendelian diseases, you expect that the effect size is very large, but the alleles are very rare. For for common diseases, it fits with the common disease, common variant hypothesis, where you would expect a common variant to have a very, very small effect. And these are studies that target these kinds of effects. While there have been a few success stories, these studies need huge amounts of patience and a ton of money, and we're not very mechanistic. So people would identify SNPs associated to a given region. In some cases, they had biological plausibility, but usually these SNPs are not in a gene, and we have no idea what the actual SNP, what the role of the SNP is in the disease process. So they're not trendy right now, but there have been a few successes with GWASs. And in 2005, now we get to the next generation sequencing, or séquençage à double in French, which has completely also been a revolution in clinical genetics. So the principle varies, and some of you may be very aware of that. It varies from platform to platform. This one was from Illumina. The basic principle is the same. So you build you share DNA, build a library. In this case, you link an adapter, put that on plates where you amplify a segment, and then they sequence but in parallel massively with different techniques, usually still using the Terminator dye, the Terminator, and then you align the sequences. Now, this has had a significant impact on cost and productivity. So you see that in 2000, at the time of the Human Genome Complex, the Human Genome Project would be about $100 million per gigabase. We're now down to nearly nothing. Maybe the technique would right now. If you want to do an exome sequencing, it would probably cost about around $1,000 if you do a reasonable number of them. And the costs is still coming down, and the output has increased significantly. So this has completely revolutionized how you approach genetics in the clinic. And I'll also get to that later. Obviously, there are a few limitations. One of which is that the errors are more frequent than with typical Sanger sequencing, typically. But now that the output and is so high, you can do 30x, 40x, 80x, 120x coverage. It greatly reduces the number of errors, but it's still possible. In your early application stages, you introduce errors. This is also improving continuously, but the length of the reading length is much shorter, so 30 to 400 base pairs, whereas Sanger is more in the 500 to 1 kilobase range, which makes it hard to detect these repetitive regions, especially these triplet repeat diseases, and also makes it hard to sequence a new sequence for an organism, because it's hard to align these sequences. And the last one is not insignificant. It's a nice problem to have, but it's still a problem. Is that these generate a huge amount of data. So if we imagined we estimate the human genome to be about 3.7 billion bases. So if we do the exome, it's 1-2% of that. But if we want to do a whole genome, eventually we'll get to that. It generates a huge amount of data, for which you have to make a selection and find what's significant. So analysis is probably harder than at the actual sequencing. But I'd say certainly harder. And here is one relatively recent example of using exome sequencing in a project. So they were looking at 84 patients with progressive myoclonic epilepsy.